We have the pleasure of having today as our speaker, Niamh Mimnagh. She received a BSc in Applied Maths and a Master's in Data Science and Analytics from Maynooth University. I had the joy of working with her during her Master's, and now I have the joy of working with her during her PhD. She's now a PhD researcher, currently in her third year in the Center for Research Training and Foundations of Data Science, working with estimating animal abundance. So, Niamh, the floor is yours. Thank you very much for accepting our invitation.
Perfect, thank you. Hi, everybody. So, like Raphael said, my name is Niamh Mimnagh, and I'm in the third year of my PhD, and it's a part of Science Foundation Ireland's Centre for Research Training in Foundations of Data Science programme. I'm based in Maynooth University in Ireland, and I've been working with Raphael on some different methods for estimating animal abundance. And today I'm going to talk to you about one of those particular methods, which is a multi-species and mixture model, and how we might use that to estimate abundance in animal communities. My thing's not working, hold on. Oh, there we go. Okay. Before I get on to talking about the mixture models, though, I like to start by talking for a bit about why they're so important. Why being able to obtain an accurate estimate of animal population size is so important in areas like conservation and wildlife management. If we go way back, sort of in the past 500 million years, the Earth has experienced 5 major mass extinction events. And these major mass extinction events are events during which at least 70% of all animal life on Earth has died. And it is strongly speculated that we're currently experiencing a 6th major mass extinction event. In just the last 500 years, an average of 6 species has become extinct every 10 years. And that might not seem like an awful lot, but that's just the ones that we know about. Being able to obtain an accurate estimate of abundance is really crucial if we want to be able to tell when species populations are starting to decline. And that's just one situation in which we need an accurate estimate of animal abundance. There are others as well. There are certain species whose populations might need to be controlled if they start to become too large. But for the majority of species that don't require conservation or control, their populations just need to be monitored. And in all of these situations, having an accurate estimate of their abundance is really crucial. There are lots of different ways of collecting data on animal population sizes. And the one I want to talk to you today is about counts of individuals. And that's exactly what it sounds like, I suppose. A researcher goes out into an animal's habitat for a certain period of time, and they just count the number of individuals of a certain species that they see or hear while they're out there. Now, this is a really attractive method for collecting data because of how inexpensive it is to carry out when you compare it with some other methods of collecting data, some other sort of more direct methods that involve trapping and things like that. So it's really an attractive method, but it does have one major drawback, and that is an issue that is called imperfect detection. And now imperfect detection refers to the fact that the number of individuals that are observed at a certain site and the number of individuals that are actually at that site are not necessarily going to be the same. This can be for sort of any number of reasons. It can be like for this owl, it can be because of the animal's coloring, it can be because of their behavior, or it could be an error on the part of the observer. It could be any number of reasons, but what it comes down to is these animals don't want to be seen by humans, so your observed abundance, that is the counts that are collected during your survey, and the actual abundance, the actual population size at that site, probably not going to be the same. So while this type of data is attractive due to how relatively inexpensive it is, if we want to use it to obtain an accurate estimate of animal population size, we need to work with it in a way that takes this imperfect detection into account. And that is where N-mixture models come in. They're a really cool family of models that allow us to work with these counts of individuals because imperfect detection is built directly into the model. This is the original N-mixture model, which was proposed by Andrew Royle in 2004. We have the top level of the hierarchy, which are the Y values. And these are the observed abundances, the counts that are collected during the survey. And here the subscript I represents the site and the subscript T represents the sampling occasion. So we have a separate observed abundance for every site and sampling occasion. And these observed abundances are assumed to follow a binomial distribution, and they depend on two parameters, N and P. N is the actual abundance at that site, and P is the probability of detection. And it's this probability of detection which is how imperfect detection is incorporated into these models. Sort of, if you were in a situation where your probability of detection was low, then you would be more likely to have data that suffers quite highly from imperfect detection than you would if you were in a situation where your probability of detection was high. And then we have our actual abundance, which is assumed to follow a Poisson distribution with a parameter lambda, where lambda is the mean number of individuals at a site. Now this is what the original N-mixture model looks like. And we've been working on a few different extensions to this model. The first thing that you'll notice is that this model can only deal with one species at a time. It can't be used to sort of model whole communities at one time. And that was what we wanted to look at first. We wanted to look at extending this model so that we could look at multiple species simultaneously, which would allow us to model sort of whole communities. Now there have been previous multi-species N-mixture models proposed, but one thing in particular that we wanted to look at was we wanted to look at estimating the abundance of multiple species simultaneously and also estimating the correlation between abundances of different species. So that's where we started with our multi-species N-mixture model. After that, we looked at a model that would allow us to work with data that was collected over really long time frames. And after that, again, we looked at a model that would allow us to work with data that contained a large number of zero counts. But I will start with our original multi-species N-mixture model, which looks like this. So we have our observed abundance, which follows a binomial distribution, and our actual abundance, which follows a Poisson distribution, which is the same as the original N-mixture model. You'll see that now in our observed and our actual abundances, we have an extra subscript S. S represents the species that's being examined. And we are able to model multiple species simultaneously and to estimate the correlations between abundances of different species by allowing the rate parameter of our Poisson distribution lambda, by allowing that to depend on a random variable that we call A. And we let A have a multivariate normal distribution. So it has two parameters, a mean vector mu and a covariance matrix sigma. And it is because A follows a multivariate normal distribution with this covariance matrix sigma that we are able to estimate the correlations between abundances of different species. This is a sort of simple example of what a correlation matrix from this model might look like. in a situation where we have 10 different species. So here each row and column of the matrix corresponds to one species. Now, what estimating these correlations lets us do is it lets us go beyond just estimating the abundance of the species, and it lets us start making inferences about the relationships that these species have with one another. For example, if two species abundances have got a very strong positive correlation, then what that suggests to us is that as the abundance of one species is increasing, the abundance of the other species is also increasing. And likewise, if the abundance of one species is decreasing, the other species' abundance is also decreasing. So a strong positive correlation tells us that the abundances of these species seem to increase and decrease together. On the other hand, if you've got a very strong negative correlation, what that suggests is maybe that as the abundance of one species is increasing, the abundance of the other species is decreasing. And I guess if your abundance is very close to zero, it possibly suggests that these two species might not have very much to do with one another. So that was our first multi-species and mixture model. Next, we wanted to look at working with data that was collected over several years, so long time frames. One of the assumptions of the N-mixture model is that the populations are closed with respect to births, deaths, and migration. And what that means is we're assuming that during the time that the data was collected, that the population sizes didn't have a chance to change due to births, deaths, or migration. And this is obviously a really restrictive assumption in a real-world setting. It restricts us to just data that was collected within a very short time frame. If we want to look at data that is collected over longer time frames, we need to work on relaxing this assumption. And that's what we wanted to do. We wanted to work with data that was collected over several years. So we started working on an autoregressive model, which is this one here. So you'll see that in our observed and actual abundances, we have another subscript again. K represents the time that the data was collected. In our case, we wanted to work with data that was collected annually for several years, so we call K the year that the data was collected. We are able to relax the assumption that the population is closed through the addition of an autoregressive term on the abundance. So what this third line is saying is that we're allowing the mean abundance at time K to depend on the actual abundance at time K minus 1. So we have phi of s, which is our autoregressive parameter, and then we have this sort of awkward looking term, log of n at K minus 1 plus 1. Originally, we were trying to work with this sort of simpler parameter, so phi of s times n at K minus 1. What we found though was that as time went on, as K became larger, we were getting really unreasonably large estimates for the abundance. We were getting abundances in the hundreds and hundreds of thousands. So to stop that happening, we are instead using the log of n at K minus 1. And then the plus 1 term is to make up for when n at K minus 1 was equal to 0. And then we have our sort of, as well as our autoregressive term, we have A, which is still our random variable, which is how we estimate the correlations between abundances of different species. Now this is an AR1 model. The abundance at time K depends only on the abundance at time K minus 1, but you could easily add more autoregressive terms to the abundance if you wanted to allow it to depend on the abundances at times k minus 2, k minus 3, and so on. So that was our autoregressive model, which was the second model we were working on. After that, we wanted to look at working with data that contained large numbers of zero counts. And for this, we started working on a Hurdle-Poisson model. And a Hurdle-Poisson distribution is one that can be used within the N-mixture model instead of the Poisson distribution when you're in a situation where the Poisson distribution doesn't fit due to an excess of zero counts. And this is a really, really common situation to encounter when you're working with counts of individuals. It's really, really common for large proportions of your data set to contain no sightings of any individuals at all. There are, there are two main methods of dealing with zero-inflated data. And I'm just going to talk about them for a second before I go on to talking about our hurdle model, just for anybody who might not be super familiar with it. So there are two main methods that I would be familiar with anyway. And these are a zero-inflated model and a zero-altered model, which is also called a hurdle model. These both belong to a family of models called two-part models. And they're called two-part models because they allow for two different processes to take place. They have a zero process, which determines whether your count is zero, and they have a counting process, which determines the value of your sort of non-zero counts for the most part. This is, sorry?
I think John has a question. He just put his hand up.
Oh.
Okay.
Oh, I'll save it till later.
Oh, sure. Okay. Sorry, Niamh.
This is an illustration that's normally used for these different types of models to explain how they work. So I'll start with the zero-inflated model, which is on the left. In this situation, we have data that contains a large number of zero counts. And what's important in this situation is that we care where those zeros are coming from. We call them true zeros and false zeros. And a true zero is a zero that occurs because the animal was genuinely not at that site, because the site is not an appropriate one. And the false zeros are every other kind of zero. They're zeros that occur because the animal was at that site, or but they weren't seen. In this case, it says sort of, I was here, but you didn't see me, I was under the water, or you thought I was a crocodile, or things like that. So in the case of the zero-inflated model, we have our zero process, which is on the top, and that produces only that produces only the false zeros. And then we have our count process on the bottom, and that can produce positive counts, but also the true zeros. And then on the right, we have our hurdle model, which is the one that we were interested in. And at least in my mind, it is a simpler model to understand, because again, we're in a situation where we have data that contains a large number of zero counts. But in this situation, we don't care where the zeros are coming from. They could be true zeros or false zeros. It doesn't matter. All that matters is that they are zeros, so we just clump them all in together. So that's what this illustration on the right is telling us. We have on top is our zero process, and that produces all of the zero counts. And then on the bottom is our count process, which can only produce positive counts. And that is the difference between a zero-inflated and a hurdle model. In a zero-inflated model, your count process can produce zero counts, but in a hurdle model, your count process can only produce strictly positive counts. So that's just for people who might not be incredibly familiar with hurdle models. This is what our hurdle model looks like. it has got two components that correspond to that two-stage data generating process. We have our zero process, which here I've called the occupancy process, because if it determines that the count is 0, then your site is unoccupied. And if it determines the count is greater than 0, then your site is occupied. And this is assumed to follow a Bernoulli distribution with a rate parameter theta. Theta is the probability of obtaining a positive count. Now these are called, these are called hurdle models, because if your zero process determines that your count is non-zero, that your count is positive, it's referred to as the hurdle being crossed, and now counting can begin. And then we have our counting process, which we assume follows a zero truncated Poisson distribution. And this zero truncated Poisson distribution can only produce positive counts. This cannot produce zeros. If instead we were using a Poisson distribution here in our count process, then that would be able to produce zeros, and we would have a zero-inflated model instead. And then in this case, our actual abundance n is equal to 0 if our zero process, the occupancy process, is equal to 0, and it's equal to the value of our count process if our occupancy process is equal to 1. And then the rest of this model is similar to our multi-species and mixture model. We again have our random variable A on the abundance, which allows us to estimate the correlations between abundances of different species. And finally, we have what we've called a hurdle AR model, which is a straightforward combination of the hurdle model and the autoregressive model. We've got our autoregressive term in the abundance from our autoregressive model, and then we've got our two-stage data generating process from the hurdle model. And this type of model is useful if you're in a situation where you have data that was collected over large time spans that also contains a large number of zero counts. So they were the 4 models that we were working with. Once we came up with them, we wanted to make sure that they were doing what we thought that they were doing, and that they were producing accurate estimates of the abundance, the interspecies correlations, and the probability of detection. And we also wanted to make sure that our models worked at different scales. So we ran a small scale simulation study and a large scale simulation study. Our small scale simulation study, we simulated data that came from 10 sites and was associated with 5 species and 5 sampling occasions. And for our large scale simulation study, we had 100 sites and 10 species and 10 sampling occasions. For each of these small and large scale simulation studies, we also wanted to see what would happen if we varied the detection probability, the size of the abundance, and in the case of the hurdle models, the degree of zero inflation in the model. So simulations that had a small probability of detection had probability of detection between 0.1 and 0.4. Large detection probabilities were between 0.5 and 0.9. Then for the abundance, small abundances lay between 0 and 50, and large abundances lay between 50 and 700. And in the case of our hurdle models, we said that a small degree of zero inflation had 20% of the data set equal to 0. And a large degree of zero inflation had 50%, half of our data set equal to 0. For each of these situations, we produced 100 data sets. So we ended up with a simulation study that contained around 4,000 data sets. the accuracy of the estimates for the interspecies correlations and the probability of detection we determined using relative bias. And this is the formula for relative bias on the top. This says that the relative bias for a certain parameter theta is equal to the estimated value of theta minus the true value divided by the true value. And the smaller the value for the relative bias that we obtained, the closer we could say that our estimated values were to the true values. And then for the estimates of our abundance, we used what's called the concordance correlation coefficient. And that is given by the formula on the bottom. Now in this formula, we have mu and sigma, which are the mean and standard deviation of the true values for the abundance. And then mu hat and sigma hat are the mean and standard deviation of the estimated values of the abundance. And in this formula, rho is Pearson's correlation coefficient. And the reason we used the concordance correlation rather than Pearson's correlation is Pearson's correlation is invariant under changes in location and scale. So what that means is that Pearson's correlation will tell you if there's a linear relationship between your two data sets. But if they have got a linear relationship, but they're very different in terms of their location or scale, Pearson's correlation coefficient won't tell you this. But because the concordance correlation sort of mixes Pearson's correlation with the mean and standard deviation of both of the data sets, if your data sets are different in terms of their location or their scale, the concordance correlation coefficient will pick up on this. And the larger the value, the closer the value is to 1 of the concordance correlation, the closer our estimated abundances were to the true abundances. Okay, for the results of the simulation study, I'm just showing you the results of the hurdle model. The reason for that is the results for the original multi-species and mixture model and the autoregressive model looked almost identical to the results of the hurdle model when we have small zero inflation. So that's these plots on the left. And that makes sense, I guess, because the data that we used for the multi-species and mixture model and the autoregressive model was not zero inflated. So to save you all from having to look at the same plot 3 times, I've just gone with the hurdle model results. Remember, in our hurdle model, we were varying 3 things in our simulation study. The first was the degree of zero inflation. So here our 2 plots on the left have got 20% of their data set was equal to 0. And on the left, we have half of the data set that was composed of 0 counts. And then we were also varying the size of the detection probability. So we had small detection probability that was between 0.1 and 0.4, and then large detection probability between 0.5 and 0.9. And we were also varying the size of the abundance. So we have small abundance and large abundance. In these plots, the yellow points are the concordance correlation coefficient for our estimates of the abundance. The blue points are the relative bias for our probability of detection. And the red points are the relative bias for our interspecies correlations. So what we want is concordance correlation coefficients that are close to 1 and relative biases that are close to 0. So what we can see first is that across the board we have concordance correlation coefficients that are between 0.97 and 0.99, which is good. It means that we're producing pretty accurate estimates of abundance in all situations. The estimates of abundance are better in each case when the probability of detection is large, which makes sense when you think about it. The larger the probability of detection, the more likely that more of the animals that are present at a site will be observed. So the easier it makes it to estimate the actual population. Then if we look at our blue points, which is the relative bias for the probability of detection, we also see that probability of detection is estimated better when probability of detection is large, which I think also makes sense for the same reasons as I said for the concordance correlation coefficient. Finally, we have our relative bias for our interspecies correlations, which is our red points. Changing from small to large probability and abundance doesn't seem to have any effect, really at all, on the estimates of the interspecies correlations. But what does have an effect is going from a small amount of zero inflation to a larger amount of zero inflation. When we go from small zero inflation to a larger zero inflation, it increases the relative bias for our interspecies correlations, which means that our interspecies correlations aren't being estimated as well in this situation, which also makes sense. If you the more of your data set that is composed of zero values, the less data you have with which to estimate your correlations. And finally, for these results, we found that the best situation for the hurdle model, the situation that produced the most accurate estimates across the board, was when we have a small degree of zero inflation, but both our probability of detection and our abundance is large. This is the situation that produced very small relative biases for our detection probability and correlation, and a high concordance correlation coefficient for our estimates of the abundance. So once we finished our simulation results, we had a quick look at a case study. And we were looking at data that was collected as part of the North American Breeding Bird Survey. And this is a large-scale survey that's been collected annually since 1966. And it involves the collection of data on 500 bird species, more than 500 bird species, across thousands of sites in the US and Canada. Each of these sites is around 24.5 miles long, and it's composed of 50 different stops. And the way that the survey is carried out is once a year in June or July, surveyors go out and they spend 3 minutes at each of the 50 stops in a route, and they record the number of individuals of whatever bird species that they see or hear while they're out there. In our case, we wanted to look at data that was collected in Alaska between 2010 and 2019. So these are the sites in Alaska here. Alaska has 94 different sites. Alaska also has more bald eagles than any other site, any other state in the US. The bald eagle population in Alaska actually accounts for half of the global population of bald eagles. And bald eagles are cool. So we wanted to have a look at what was going on with the bald eagles in Alaska. So we chose them as one of the species that we really wanted to look at. But there's also more than 200 other species that appear somewhere in Alaska between 2010 and 2019. So we needed to come up with a subset of these. We chose to look at 10 species overall, and we wanted a mix of species in terms of their habitat and things like that. So in this case, we have 3 other species that are water birds, like the bald eagle. They are the trumpeter swan, Wilson snipe, and Canada goose. So these are all species that you would expect to live along the coastline, sort of have similar habitats to the bald eagle. And then we have a selection of other species that can be found. So some of them live on the coast and inland, and some of them are found really mainly inland. We wanted sort of a bit of a mix so we could see what was going on. So these are the 10 species that we decided to look at. So we're looking at data that was collected between 2010 and 2019. So that's 10 years of data. So going into this, we assumed that the population sizes would have changed during this time, that the population sizes would not have remained static within these 10 years. So we assumed going into it that a model that allowed for population sizes to change, our autoregressive model, or one of the models with an autoregressive component, would be a good fit for this model, for this data. What we also found was, when we put our data set together, was that 80% of our data set was composed of zero counts. So that led us to think that maybe we might also need our hurdle components in the model. So we went into this thinking that, okay, the hurdle AR model might be sort of the best out of the models that we have to offer. We fitted each of the models, so the original, multi-species, and mixture, the autoregressive model, the hurdle model, and then the hurdle autoregressive model. We fitted each of them to the data. And another thing that we wanted to look at was the dimension of the probability of detection. So you can have a probability of detection that sort of is different for every species. So in this case, we would just have 10 different probabilities of detection. Or you can have it that's different for every species and site, or for every species, site, and year. So we looked at all of these different combinations. We fitted each of the models with the 3 different combinations, probability of detection that varies by site, species, and year. Then we looked at site and species, and then just species by itself. We also fitted each of the models with and without covariates on the abundance. The covariates that we had on hand were the latitude and longitude of the sites. So we fitted sort of each of these combinations. I can't remember how many combinations it was in the end, 18 maybe. And we compared them using DIC values, where DIC values are a generalization of AIC values that are useful in a hierarchical modeling situation. And what we found was that both when we had covariates on the abundance and when we did not have covariates on the abundance, the hurdle AR model was the best fit. This is what we thought going in. The difference was that when we initially fit our models without lat long on the abundance, the model that was chosen as the best one, because it had the lowest DIC value, was the one where we had a probability of detection that varied by site, species, and year. But when we added our covariates to the abundance, this changed. So now the best model was the hurdle AR model, where probability of detection just varied by site and species. So I guess we could say that the variation that was initially explained by this extra dimension on the probability of detection was now being explained by the covariates that we had on the abundance. Overall, the model that had the lowest DIC value was the hurdle AR model that had the covariates on the abundance and the detection probability that varied by site and species. So that is the model that sort of we went on and looked at the results for, and that's the one that I'm going to show you here. Like I said, we were pretty interested in the bald eagle in particular, So there, it's the results for the bald eagle that I'm going to show you here. But you could look at any number of species. You could spend ages looking at all these different species. What we found was that for the bald eagle, of the 94 sites that were available in Alaska, the bald eagle population is concentrated at just 18 sites that are along the southeastern coast in a series of islands that are called the Alexander Archipelago, which was interesting. I mean, you would expect that maybe bald eagle populations would not be thriving inland, so you wouldn't expect them in sort of any of these inland sites. But it was interesting that they are not present at any of these other coastal sites. They're really just along the Alexander Archipelago. So once we noticed that, we zoomed in on just these 18 sites, and this is what we found. So this is just the 18 sites along those islands, and we've got the abundance for bald eagles by year. And what we noticed was that while some of the sites sort of remain quite low, they have quite low populations, there are some sites where the bald eagle population was growing every year. So this very bottom site in particular goes from having basically none to around 30 between 2010 and 2018. And there's a few other sites as well. I think this one, I'm hoping you can see my cursor, but yeah, what we thought was that there might actually be population growth happening in this area during this time. So as a sort of preliminary kind of investigation into this, we looked at the mean bald eagle abundance in just this area by year. And this is what we found. So along the x-axis, we have the year, and the y-axis is the mean abundance. The blue line is the mean abundance, and the ribbon is a 95% credible interval. So what we found was that, I mean, looking at this plot, it does seem that there was a general increase in bald eagle population size in this area, definitely between 2010 and 2018. There was sort of some little dips, but it did seem to be like a general upward trend, which was really interesting. But then something happened in 2019, and we're not actually sure what. We're waiting on the new data. I know that it probably wasn't, they probably didn't collect data last year, so we might be waiting a little while. But I would like to see sort of if there was a sharp drop off in 2019, and we would like to see, it'd be interesting to see, was there something happened? Does it continue to decline, or is there something going on, or was that sort of quite a sharp dip in an otherwise increasing population? We're really interested to see whenever the new data comes out. I'm waiting. And finally, we had the correlations between the abundances of the 10 species that we looked at. And the thing with these correlation matrices is that there's so much going on. You could really zoom in and look at the relationships of all of the species, and you could be here forever. Especially like the Canada goose, which seems to have some quite strong relationships with other species. Like it's got quite a strong negative correlation with Sir Swainson's thrush and the red-breasted sapsucker, but sort of quite a strong positive correlation with, which one is this? Wilson's snipe. Oops. So like the correlations are interesting. I think it would be really interesting for someone who has more of an ecological background than I do. My background is sort of maths and stats, to sort of tell us what they think about what's going on with these species. But for the sake of our one, we were again mainly focusing on the bald eagle. And sort of what you can see is that, so the bald eagle is here. It's got a positive correlation with Canada goose, Wilson snipe, and the trumpeter swan. And these are the 3 species that also live on the coastline. These are the other water birds. So we thought that maybe that might be a reason for the positive correlation, that maybe their abundances might be dependent on similar factors. It's also got a strong positive correlation, well, a positive correlation with the tree swallow. And a tree swallow can be found all over Alaska. They live on the coast and inland, so this could be something. And it's got a negative correlation with Swainson's thrush, the red-breasted sapsucker, and Hammond's flycatcher. And these are birds that are mainly found inland. So we thought that that might be a reason for that. But like I said, this would be really interesting for someone who has an ecological background. I'd be really interested to see what people thought about it. But that is everything from me.